So we're just going to run through the light-dependent reaction. So all of this is taking place inside the chloroplast. That's our organelle in the plant cells. The stroma is the fluid inside the chloroplast where everything is suspended. This structure here, this is our thylakoid. Photosynthesis takes place inside these proteins in the membrane of the thylakoid, membrane. Their first stop is going to be Photosystem 2, or P680, or non-cyclic photosystem. Okay. The first stop, what do we need? We need carbon dioxide and water. This is going to be for the Calvin cycle, so we don't need to worry about that right now. This is going to come from our soil, and it's going to go to photosystem 2 first. So let's write that in. So we have water, and it's going to come to photosystem 2, and then what's going to happen? Chlorophyll inside photosystem. How do I write this? So the chlorophyll inside the photosystem is going to collect sunlight and use that energy to split the water. If you remember, this process was called photolysis, using photons, using sunlight to cut something, in this case cutting water. So water comes in, it's cut, the electrons are excited there, and they split. We have oxygen and hydrogen protons. This oxygen is going to leave the plant, and that's the oxygen that we're going to breathe, so it's very important. These hydrogen ions, for all intents and purposes, are going to float here. Okay, they are going to chill in the thylakoid lumen. That is the fluid inside the thylakoid. Okay, this is the stroma, this is the lumen. So water comes in, chlorophyll collects sunlight, and it's going to cause it to split. What happens next? These electrons are going to make their way up photosystem 2. And when I say make their way up, that is to say that they are gaining energy. Okay, we have a series of redox reactions that are happening in here, and it's going to cause those electrons to gain energy. Once they gain energy, they're going to come over and get transferred to, I'll be consistent, to the next structure. So this is our cytochrome complex. Remember, these are all embedded in the thylakoid membrane. So our electrons arrive, they come in, and now they're going to go down what we often depict as this ladder. And what this is our electron transport chain. So we are going to lose energy, but this process is very important. By losing energy, let's use purple. By losing energy, we are going to help hydrogen ions move across their gradient into the lumen. So we lose energy, we bring hydrogen ions across from the stroma down into the lumen of the thylakoid. And now these electrons have a little bit less energy than what they had after being excited by photosystem 2. Great, so the next destination is to get passed on to the last protein here. This one is called photosystem 1, or P700, or the cyclic photosystem. Like before, we have chlorophyll here. We have chlorophyll. It's going to collect sun. It's going to excite the electrons, and those electrons are going to go, quote unquote, up to the top of photosystem 1, where they will be picked up. What is going to pick them up is NADP+. We call this an electron carrier. You'll note that it has a positive denotation. If it's positive, it means it is ready to accept something negative. It is ready to accept an electron and form a bond. Now, at this point, all over the stroma, we have hydrogen ions everywhere. They're just floating. Okay, so when we pick up these electrons, one of these hydrogen ions are going to come in and help make that bond, just to keep things tidy. So then we get NADPH. Those electrons, they're going to be over here. They're going to help make that bond. Okay, and so you might say that at the end of the day, these electrons ultimately came from water. You will note, however, there is no photolysis here. Okay, that is only in photosystem 2, not photosystem 1. So our electrons keep going through. They go through the electron transfer chain. They pull hydrogen ions across. So they pull hydrogen ions across. They pull hydrogen ions across. So that's increasing. As we keep using more water, we get more hydrogen ions coming in. Okay, this is from the H2O. So now, at some point, we're going to have more hydrogen ions inside than outside. Okay, maybe better if I say number. So the number of hydrogen ions are going to exceed those outside. What is this going to do? It is going to establish a voltage. It is going to establish an electrochemical gradient. An electrochemical gradient. So now it is more positive inside than in the stroma. Remember, this is all in the lumen. So it's more positive inside the lumen than it is outside in the stroma. So they're going to go somewhere. They're going to go down, travel down the gradient from high to low. Okay, they want to move from high concentration inside the lumen to low concentration in the stroma. And while they do that, they're going to pass through this structure called ATP synthase. So when they pass through, let's say all of these hydrogen ions go through. All of these hydrogen ions, they go through and they pop out the other side. this process of going down their gradient, across their voltage gradient, is going to be called chemiosmosis. Now, when they do that, they are going to lose energy. They're going to lose some of that potential energy, some of that voltage while they pass through. So when that happens, we are able to make an energy-rich ATP molecule. So ATP, if we had like an A attached one phosphate group, two phosphate groups, that's triphosphate, because we originally started with a DP, diphosphate, plus phosphate. So we use this energy to create this molecule. This process is called phosphorylation. Phosphorylation. Therefore, you might say, in brackets, because this process ultimately uses energy that was generated by the sun, that it was photophosphorylated, or photophosphorylation. So that is the light-dependent reaction in the thylakoid, primarily in the thylakoid lumen and the thylakoid membrane. The next step is to take the ATP that we generate here and pull it to the side, all this NADPH, pull it to the side, all of that CO2, pull it over here, and start the Calvin cycle. This is where we actually fix the carbon into the sugar rings. But we'll do that in the next video.